Mr. Hyatt here. This is the AP Biology Chapter 4 lecture. As you can see on the screen, we're continuing our investigation into the world of the chemistry of life. And we're talking about carbon and then carbon-based molecules. So there's a lot of potential memorization in this chapter. So make sure if you're a Quizlet person, look for that resource. If you're a flashcard person, make your flashcards. Whatever strategy works for you, great. Organic chem is the branch of science that studies carbon-based compounds. Also is the pre-med weed-out class in most colleges, so you got that to look forward to. So carbon forms 4 covalent bonds just because of the number of valence electrons. It's tetrahedral, so it's going to be cross-shaped, kind of. Bonds really easily to itself because it's going to share covalent bonds, it's going to share electrons, it's going to stabilize itself. So major elements of organic molecules, of course, carbon, hydrogen, oxygen, nitrogen. So that should sound familiar. Those are the 4 elements that are essential for life. Those are the 4 macro elements: CHON, carbon, hydrogen, oxygen, nitrogen. And you see there the valence electrons and the charge that accompanies those when they form bonds. So the valences, the number of valence electrons, those are the rules for building organic molecules. So we can only build as many as many over 4 electrons or as many under 4 electrons as we have in the valence shell. So hydrocarbons are going to be things made up of only carbon and hydrogen. Something like methane being an example. C2H6 would be an example. C2H4 would be an example. So those are molecules that only have those 2 elements. No nitrogen, no oxygen. There's a couple of examples. Methane, ethane, and ethene. Those are the 3 that I referenced. So you can see they're only carbon and hydrogen. Another vocab term, isomers are things that have the same molecular formula but different structures. So again, like we said earlier on in one of the other chapters, form dictates function. So the shape is going to control what it does. Let's take a look at some of these. Notice top right, bottom left, those are isomers. Same structure, same composition, different shape. Different shape, different function. There are 3 types of isomers: structural, geometric isomers, and enantiomers. We'll talk about each of those coming up on their own slide. So structural is just going to be different arrangement of their atoms. So we saw one right here. We're rearranging the atoms, so gives it different properties. Rearrange the atoms here, gives us different properties. Here we've got where we're moving our double bond. A geometric isomer, same covalent partnership, but different spatial arrangement. So that's going to be more closely related to those double bonds that I showed you in the other one. And here's a picture. So a structural isomer is here. Geometric isomer. Okay, so here's kind of the key. Look where our functional groups are, our X's. We're going to talk a lot about those towards the end of this chapter. So we've got a cis molecule here where they're on the same side. We've got a trans molecule here where they're on opposite sides. If you're socially conscious, you've probably heard the term cisgender and transgender. Cisgender refers to someone who identifies with the same gender that they were born with. So they're on the same side, cis. Trans, they're on opposite, so someone that identifies with the opposite gender that they were born with. So in that case, society took a cue from chemistry, and that's where those came from. Next up is enantiomers. They're mirror images of each other. They're going to usually involve some sort of an asymmetrical carbon. We're going to have some kind of a funky carbon-based side chain, something along those lines. We often refer to these as left-hand and right-hand enantiomers. So you see here, the thumb is the NH2. Same thing here, but it's left hand, right hand. So they're mirror images of one another. So organisms are incredibly sensitive to even the smallest changes in shape. Got an example here. Thalidomide There are two isomers. One is an effective drug that treats a number of things, and the other causes birth defects. One is okay for mom to have, the other is not. So we need to be very conscious of which version of the molecule that we're ingesting, that we're releasing into the atmosphere, all those types of things. This is where some of the memorization is going to come in. You just to be able to function in organic chemistry, you have to be able to speak the language, and unfortunately, a lot of the language is memorizing the shapes of common groups of elements. Rather than having to memorize a billion different molecules and what they do and how they do them, we memorize a few functional groups, and then when we see those things, we can say, oh, that's a ketone. That probably does these 5 or 6 things. Oh, that's an alcohol. That probably does these 5 or 6 things. So it's kind of annoying to have to memorize 15 to 20 things, but it's better than having to memorize a million or even more. Kind of an example of functional groups. Notice the difference here is that we've got a double bond between testosterone and estradiol. That's a permutation, a different version of estrogen. It's a female chemical and a male chemical. We've got a different functional group here. We've got a different functional group here. Changes our number of double bonds and changes the functional group that attaches there. So they're going to be really important to the function of an overall molecule. So as we look at these functional groups, focus on the structure, look for things that are unique to the functional group, then that will help you connect to the properties, and then hopefully you can identify or name some examples of molecules with that functional group. Starting off, a hydroxyl group that's an alcohol. You see there's an example of our ethanol that's present in alcoholic beverages. You see some of the properties there. It's polar, forms hydrogen bonds with water molecules that dissolves organic compounds such as sugars. That's going to be really important someday when you grow up and you can ingest these types of things. Alcohols dissolve sugars, so that's one of the reasons that it manipulates how you feel, and particularly how you feel the next day. Carbonyl groups, we're looking at this carbonyl is going to be a carbon-oxygen double bond. We got ketones and aldehydes. There are two types of carbonyl functions, and you can see what their structures are. Again, if you're a flashcard person, this is an awesome chance at that. If you're a Quizlet person, I've got a reference to that, a link to that on Canvas. So here's the aldehyde. Sometimes we write that as CHO. Again, it's a carbonyl group at the end of a carbon skeleton. A ketone is different in that the double bond is in the middle of the carbon chain. So still CO double bond, it's just an aldehyde is at the end, a ketone is at the middle. If you think about what's going on with the electrons here, we're at the end of a carbon chain double bond. So what are we going to have with electrons out here versus what are we going to have with electrons here? Again, form dictates function. So we got to understand how the shape of that molecule and the location of that double bond is impacting the overall molecule. Carboxyl groups are kind of like carbonyls, except they've got an alcohol stuck on the end, or a hydroxyl stuck on the end. It's going to be acidic because the covalent bond between oxygen and hydrogen is so polar. So you see there, acetic acid is an example. Gives vinegar that sour taste. So yeah, that's a carboxyl. Carboxylic acids are going to be things that donate hydrogens. They have carboxyl groups. They form a lot of weak organic acids. Acetic acid is one of those. Amino, those are easy because they have nitrogen in them. We're looking for amines. We're going to see amino acids and all sorts of different versions of those. But those are kind of an easy one. They were for me as a student anyway, because I just looked for that nitrogen. Sulfahydryl, I think, kind of ties into or goes right along with, I guess I should say, the aminos, because you're looking for your sulfur, and it's hard to miss that one. So there's our sulfahydryl group. I know I'm going through this quickly, but it's memorization. So me talking about them all day is not going to help you understand them any better. Phosphates are going to be humongous when we get to cell membrane function, and that's what you're kind of seeing here is a glycerol. That's a phosphate attached to a whole bunch of carbons and hydrogens. Does a lot with our basically forming our cell membranes. Moving on, our methyl groups are going to be CH3s that are stuck anywhere. Really, they're going to be side chains. But that's going to be really important. We're going to talk about methylation of DNA that that changes which genes are expressed and which ones are not. Sort of has to do with dominant and recessive, kind of. Like I said, we'll talk a lot about that, but that won't be until the spring. So we just have to kind of commit this stuff to memory so that as we move through the semester, we can call it out. And when we talk about a methyl group, we know what we're talking about. When we talk about a sulfohydryl group or a phosphate or an amine, we know exactly what we're talking about. We're all on the same page. So in summary, recognize those isomers, know what the difference between the three types of isomers are, and then know those functional groups, practice them, and be able to describe their properties and hopefully give some examples. That's chapter 4.